Hello, and welcome to another episode of Pipetting Hot Tea. I'm your host, Amelia.
I'm Aditi.
And I'm Taryn.
In today's podcast, we're digging into the fascinating world of movement, something we all do every moment of our lives without even thinking about it. Think about walking across the room, reaching for a glass of water, or even typing on your phone. All of this is powered by the nervous system.
But here's the twist. Much of that symphony is not conducted by your brain. Deep inside your spinal cord are intricate circuits that quietly orchestrate how we move. Our guest research explores how these spinal networks work and what neurons are involved.
By studying tiny zebrafish and mice and combining cutting-edge lab techniques with computational modeling, this knowledge could reshape our understanding of health, medicine, rehabilitation, and even robotics. So without further ado, please welcome Dr. Tuan Bui.
So you started in math and engineering. What led you to work in biology?
To be perfectly frank, I found engineering very boring. So when I was an undergrad, I was looking to switch fields, and it just happened that one summer I was able to find a job in the lab that was studying the spinal cord. And they knew all the biology, but they were looking to do computational simulations of the spinal cord. And they need someone with a strong math background. And so having been kind of interested in shifting fields and kind of interested in how the brain works, I thought it was a natural entry point to something different, something biological. And I was lucky enough to get the job, and 30 years later, here I am.
When people hear that the spinal cord controls movement, what should they be picturing happening inside their body?
Yeah, so people think the spinal cord has this kind of cheese string-like structure. It just sits there and it kind of passes information up and down the brain. But what happens in the spinal cord is there's thousands and thousands of neurons, and our bodies are composed of 640 muscles. So it takes a lot of neural machinery to control these muscles and to make sure that we make our movements correctly. So if you want to do something, there's commands from the brain or the brainstem that's sent down to the spinal cord, and then it will take those commands and translate it to, I have to activate these 4 muscles in this way. And so the spinal cord will take care of all of that. So if you're going to take, and you've done this with chicken, so if you're going to cut chicken's head off in a way that is still alive, it's kind of tricky, you can actually keep it walking for days and days, because all the spinal cord needs is some kind of go signal, and it's still there from the brainstem. And the spinal cord will take care of controlling all the muscles in the right way to walk. So it is very active. And that's why when someone has a spinal cord injury, depending where it's located, they lose the ability to move a lot of their muscles, because it's so important in terms of controlling each individual muscle.
Mm-hmm. And you have your own research lab, which focuses on 3 areas of research. Can you maybe explain a little bit about each sector?
Yeah. So my main interest is how the nervous system controls movement. So we kind of study the different circuits within the spinal cord and whether they control hand movement or walking, jumping, or things like that. And then I've got 2 aspects that are kind of connected to that. The first one is, when an animal is born, or when a baby is born, let's say, that a baby that's born doesn't have the full repertoire of movements that we can do as an adult. And so I'm interested in how the nervous system kind of matures to allow us to gain these new movements, because if we do that, we kind of understand how each component works. Initially, when an animal is born, it has a small number of neurons, and it can do a small number of movements. And then as it's developing, more and more neurons come online, and they're connected. And so if you can know which neurons come online later and what movements they're connected to, you have a sense of a clue as to these new neurons, they must be important for this newer, more mature movement. So there's a lot of insights to be gained about how the nervous system matures and how it gives us a better understanding how the nervous system operates as a whole. The third part, I guess, is what happens after an injury. So I mentioned after spinal cord injury, human subjects that have these injuries often lose movements forever. It is really sad. There's just very little that can be done right now to regain those movements. And so if we can somehow through our research be able to help them regain some form of movement and some form of independence, I think that would be incredibly valuable for the individuals as well as for society as a whole. So those are basically my 3 research programs.
That's really interesting.
Thank you.
All right. right, so now we're going to dig a little bit deeper into each of those research sections. So my first question to you regarding like going in deeper into this is, why is studying the maturation of motor control important, and what kinds of insights can it give us?
Yeah, sure. So I'll give you an example from zebrafish. So you may wonder, why study zebrafish and what can it give us in terms of insights? So a former PhD student, Stéphanie Coutreau, looked at a population of neurons called primary motor neurons. So when zebrafish are born, and this is pretty fascinating, but within a day of the egg being fertilized, it can already start making movements much faster than us, right?
Oh, wow, that's pretty impressive.
It is really impressive. But the movements are super simple. They're like these large body coils, because at that point, there's only a small number of neurons that exist. And one of them are called the primary motor neurons. They innervate those fast muscles that make the zebrafish coil really, really fast. And then as the zebrafish matures within the next few days, it starts making movements that are more mature. So slower swimming that actually looks like what the adult will do. And at that point, there was a second population of motor neurons called secondary motor neurons, and they emerge. So the primary motor neurons, who were kind of the bee's knees, I guess, at the beginning, they become less important afterwards. And so what we wanted to study was what makes them kind of less important as the animal matures. So what she was able to do was study different ion currents in these primary motor neurons and see that during development, they change. So when the fish becomes less and less reliant upon these primary motor neurons and develops more mature movements, these ion currents make it so that the primary motor neurons kind of take a back seat. So it's interesting because it gives us an insight as to when we mature and some neurons become less involved, there are likely to be changes in how some of these currents mediate the activity of these neurons, so they become less frequently used, and then other neurons can then take a more prominent role. So yeah, insights gained from a tiny little fish can give us a lot of insights into how our nervous system matures.
That's really cool. I want to ask you a question about your third area of research, so spinal cord injury. Most people, including myself, would assume that spinal cord injury recovery involves huge chunks of damaged tissue and rebuilding major neuron pathways, but from my understanding of after reading some of your research, you've shown that the manipulation of a single neuron population can have a huge impact on spinal cord injuries. How's that even possible?
Yeah, so you're right. I think most people do think that the best way to treat a spinal cord injury is to repair the spinal cord, for sure. You were able to regenerate the part that's injured, then that's the best. But at the same time, if you repair it, you still have to reconnect the brain to the part below the injury, and that's not easy either. What we've been kind of looking at is, in the meantime, before we develop the technology to regenerate the spinal cord that's injured, what if we use the neurons that are still there? They're there, they're just disconnected from the brain. And like I mentioned with the example of the chicken with the head cut off, right? The circuits to move your limbs are there. They're just dormant. They're just waiting for anyone to talk to them. And so these neurons that we've been studying, they take information from the skin, and then they relay it to the spinal circuits that are still there. They're just waiting for something to tell them, hey, move the limbs. And then they're able to make limbs move. Now, this is in mice, and mice are better than humans in terms of recovering locomotor activity after injury. Humans, as evolved as we think we are, we just suck at healing ourselves after injury to the nervous system. It's just the way it is, right? Like, if you do these experiments in dogs and cats, fish, they are able to, in some cases, repair the whole spinal cord or recover some form of locomotor activity. Humans, we just can't do it. We have the same neurons. We know this using molecular biology, that we have all the same neurons. It just happens that we've evolved to not be good at repairing our damaged nervous system. So we want to see if we know these neurons are useful for mice to recover locomotor activity, can we then figure out a way to then reactivate them in humans? So people with spinal cord injury, often they'll have to go through these long therapy sessions where you have a team of occupational therapists, and they're on a treadmill, and there's weight support. So their body weight is supported, and on these treadmills, these occupational therapists will take their limbs and make them walk in the treadmill. And the idea is that this kind of stimulates the sensory information in the limbs that is used to being activated during walking, and then reactivates the spinal cord, saying, hey, this is how it used to be. Let's see if we can do this again through the treadmill action. And we would like to kind of pair our understanding of how sensory neurons are really useful for that, try to stimulate those neurons, and then pair that with this kind of therapy to increase the recovery of locomotor activity. So that's the grand goal.
That's super cool.
Thank you.
Adding on to that, so like in your research, what has it revealed about like how motor circuits adapt to a movement when it goes wrong? So if a movement goes wrong, how is it adapting?
Yeah, so we have all sorts of reflexes. I think you can think of like when a doctor comes with a hammer and bangs your knee, right, and you reflexively kick or when you step on a thumbtack and you just withdraw your limb from it. So we have all these reflexes, and so they're the ones that may be useful after injury in terms of re-engaging the spinal circuits. So we're trying to basically reactivate those reflexes to make the limb move to look like walking. Now, one thing about spinal cord injury that can be actually make it even worse is that in some cases, some patients develop what's called spasticity. So that's the involuntary contraction of muscles. So imagine that you're really struggling to move your body, and now after injury, what happens is that for some reason your muscles start contracting without you being able to control it. So that makes it even worse. Sure, you're moving your muscles, but they're moving so hard and you can't control it. In some cases, it's actually painful. So what we're learning is that the spinal cord is trying to make some changes. It's either rewiring or it's growing new neuronal processes, but it's not doing it well. And in some cases, it's actually producing activity that's actually can be painful and not beneficial at all to the patient.
Okay. So is there a way to like go about like correcting that then?
Yeah. So once we understand what is going wrong, then we'll be in a better position to say, are these neurons, or is it these ion currents? Is it these neurotransmitters that are being released that shouldn't be, right? So part of the whole basic research aspect is understanding, because it's hard to treat something if we don't understand what's going wrong. We know in terms of the patient what looks wrong, but we don't understand what exactly is occurring after injury within the nervous system. So there's certainly development of certain drugs or therapies that we're trying to for example, spasticity, trying to relieve that. And I think there's some exciting avenues. We've developed in our lab a model to easily induce spasticity, so we can test different drugs. So it's something that we're certainly actively investigating, as well as trying to recover locomotion.
Okay, so like keeping all of these like different research avenues in mind, so, like can you talk us through some of the different challenges you've faced and how you've kind of like gotten past them?
Yeah, I mean, people think that some people think science is slow, right? They say, you've got this grant for X amount of money. Like, why can't you cure spinal cord injury? These experiments, we often have failed ideas. We have these great ideas and we test them, and unfortunately, we just didn't get it right. So often they're, experiments that you have the best intentions and they don't work out right. So that's one of the challenges, certainly. The other challenge is, of course, to maintain a team of researchers. I don't do much in the lab anymore. They don't let me touch anything anymore. But I certainly manage a fantastic team of researchers. So for us, it's having a great team that's assembled and also figuring out which of the ideas are the most promising for us to tackle, because we can have hundreds of ideas but limited amount of resources and time. So one of the greatest challenge, and also I think is exciting, is identifying those ideas that we think are most promising in terms of helping human patients with spinal cord injury.
Okay, so then what's your thought process on like picking like what kind of areas you want to like dig deeper into?
Yeah. That's a great idea. A great question, sorry. I mean, initially I was always interested, I've always been interested in athletics, and I've always thought about how incredible the body is in terms of just moving and the things we can accomplish. And I always felt bad for people with spinal cord injury or trauma to the brain, and they've lost that ability to easily move, right? When we move, we don't think about really. I mean, unless it's something that's novel and new, most of our movements come instantaneously almost. When I kind of pick projects, I'm really focused on kind of understanding how we move and how to recover those movements. Some things, we all have our strengths and weaknesses. So something like repairing the spinal cord, that is not my forte. I'm more interested in things to stimulate neurons or inhibit them if necessary. So the approaches we kind of look at are ways to stimulate circuits rather than repair them, just because we don't have the expertise to repair them, but we certainly have the expertise to know what neurons to target and how to best stimulate them. So yeah, it's based on interest and what we think might be successful and the approaches that we're used to using.
Okay. That's really interesting. And that's what, like, obviously neurobiology is interesting and very, for me personally as well. But what would you say is your most favorite part or interesting part in your research?
Like I said, I don't spend much time in the lab. I've got these great teams of researchers, and so I'm always excited when they present me data and to dig through it and together work through what it might mean. I might have some idea of what it means, but also my own preconceptions. And the people I work with, grad students, undergrads, postdoctoral fellows, they have their own perspective, and coming together and kind of making sense of the data. That to me, it makes me get up out of bed every morning. And sometimes my lab will hesitate to give me new data because I'll often jump to conclusions. They always tell me to moderate my excitement because A, I'm so proud of what they do, and B, just when there's good data, it just opens up so many different questions, and then that makes more work for everyone but me. So that is my favorite part for sure, is digging through the data and kind of advancing the field in whatever modest ways we can.
Mm-hmm.
Okay, so we're going to move on to our third segment that's about kind of walking in your shoes. So what a day in the life of someone in the lab or a day in your life looks like. So my first question is, I know you don't work in the lab a lot anymore, but just from your understanding of others or from your personal experiences, can you walk us through what a normal day looks like when working with live animals and what some practical realities are that people might not realize?
Certainly, yeah. So the way we work with animals in two different aspects. So in some aspects, the animals stay alive and we test them behaviorally. So for example, zebrafish, we can test how they swim. So we have these automated tracking systems where we expose them to different drugs to see how it affects their swimming and especially their motor control. So one thing that's difficult about that work is that it can be very variable. Sometimes zebrafish just won't cooperate. You'll find like in a batch of 20, 10 of them will be asleep, right? And so kind of trying to make solid conclusions is difficult if the zebrafish does not want to cooperate. It's not necessarily the drug, it's lethargic, it doesn't want to cooperate. And same thing for mice too. We also work with mice. Sometimes we put the mice on a treadmill to try to help them recover locomotion, and they're bored, they're not motivated, so that's one of the challenges with working with live animals, is that if you want to test especially their movement, you got to make sure they're motivated. If otherwise it kind of confuses the data because you think, oh, is it not working or is the animal just not caring? So that's challenging. The other aspects we do is we do a lot of neural recordings. To do that, often we have to isolate or dissect the spinal cord out. So you have to learn the surgery to remove the spinal cord, keep it alive so that you can have electrodes to then record from the neurons. I've gotten used to it. The mouse spinal cord, which is, it's about 3-4 centimeters in length. The zebrafish spinal cord is 5 millimeters in length. So trying to dissect that out is not easy. And I've tried. My PhD student, Stephanie, has tried to teach me, and I've failed miserably. And so that's part of the reason why they don't allow me in the lab, because they know I'm going to hack things away. Whereas I could still work with the mouse spinal cord, and my PhD student, Shariar, is patient with me. So those are some of the challenges is the methods of dissecting the tissue you need and to actually get the electrodes to record the activity. But once it works, it's fantastic. I mean, recording from a live brain cell, you see it fire, right? You're thinking, well, when I'm thinking or when I'm moving, that's exactly what's happening in my brain. These neurons are active, they're spiking, and I'm seeing it in a dish, and I'm recording and I'm manipulating. It gives you a sense of power, but also awe at what you can do in the lab.
Okay, so you already touched on this a bit, but like, how do you go about like transferring the animal stuff to the human stuff? Because that just seems like a big leap, especially to me. Like
Yes.
just going from like a tiny little zebrafish to like a 5-foot human being.
Absolutely. That's a great question. You know, part of why we think we can translate the findings in different vertebrates to humans is that when you look at the neurons, you can label them with different markers that tells you this is neuron A, it has this marker, and you can label it. And lo and behold, when people have looked at human tissue and used the same markers, neurons light up as well in the right spot where you think they should be. So that tells you that there's some evolutionary conservation of these neuron types. Now, I'm not going to claim that a fish moves exactly the way that a human does, right? They don't have arms, so it's but the way they move by swaying their bodies left and right. Well, when you and I walk, we don't walk on one leg on one side. We have to kind of sway from left to right all the time. And certainly they found that the neurons that do that in fish coordinate their left-right kind of body bending are the same neurons that in our spinal cord coordinate the right and left side of our bodies. So there's certain lessons that can be gained from different vertebrates to how the human nervous system works. Now, some things you can't translate. So again, you take a zebrafish, you cut its spinal cord, and you leave it there for a day, and then a day or two later, it's swimming about as if nothing were. Its spinal cord has completely repaired itself, whereas ours does not. So the value then is to say, what is it that's different in the zebrafish spinal cord in their immune response to the injury that through evolution we've kind of stopped that from occurring so that our nervous system is not able to repair itself. And there are theories as to why that is, maybe because it's become too complex that you don't allow it to repair itself because then the rewiring might be really messed up and you don't want that as a consequence. But if we can kind of understand what genes or what signals are produced in the zebrafish that have stopped being produced in the nervous system of humans, then hopefully you can kind of prod the human nervous system to repair itself again in a guided kind of way. So yeah, they seem like these animal models seem very far away from us, but there are certainly lessons that can be learned and perhaps applied to human biology.
Okay, and then in terms of like putting yourself kind of more in, I guess, your lab team's shoes, what do you do when you're faced with like a challenge? How do you go about like approaching it and like kind of like getting through those tough times?
Yeah, so like I said, we've had experiments that failed, and despite the best hypothesis, thankfully, because I have a really good team of talented and bright people, there'll be different perspectives as to why things have failed and different directions as to kind of take afterwards. And so there's always really healthy discussions. Part of the things I really enjoy is not just looking at the data, but also those discussions. Like this lab has found this. That might explain why ours failed. Or, there's this product here that I've heard can do this in this other area of the nervous system. Maybe it works for the spinal cord. So certainly, you kind of shake off the crushing disappointment of failed experiments, and then you just kind of, what else are you going to do but try again and try something new. And so thankfully, there's a lot of people around us, and we have also collaborators around the world that can kind of inspire us and guide us as to next steps to try to reach our research objectives.
All right, and now we're going to go into our last segment that we have. And this is kind of looking at your research in terms of like applying it in the future and where you think it's going to go. Mm-hmm. So I'm going to hand it over to DT.
So since we're talking about the future and everything, how do you see like your work implemented in the future? Like, are there any research areas or future pathways? Maybe like an additional research sector in your lab, something like that?
Yeah, so I'm not going to make promises to anyone listening out there that have a spinal cord injury that I'm going to cure them. But we have these neurons, these DI3 neurons, and we're working on them. We've got a team. I'm just going to name them Alex Leberté, a postdoc in my lab, Sharia Naziri, and Dexter Lashborough, are grad students. And they're looking at these neurons as possible targets for facilitating recovery of limb movements after spinal cord injury. And in the mice, we can find ways to actually activate them using light. This is, traditionally people think of neuroscience, especially the brain, using electricity, and that's very true. You use electrical stimulation to kind of excite neurons and whatnot. The problem is that it's not very precise. You have an electrode somewhere and it stimulates anything nearby. there are these advances in neuroscience where you can kind of make certain neurons express light-sensitive channels that when you illuminate with certain light of a certain frequency, only your neurons that you're targeting will be activated. So we're trying to see whether we can do that in mice so that we'd have animals with spinal cord injury, but with these special light-sensitive channels that are expressed only on neurons of interest. And we want to see in that mice that's walking, if we stimulate those neurons, even though it's spinalized, will it allow it to recover quicker? If that is the case, then we would try to find a way to kind of do the same thing in a human, where we'd find a way to selectively activate these DI3 neurons, which we know are there, but we just don't have a way to reach them right now. So I could see this is probably the most exciting for us avenue within the spinal cord injury sphere. And so we're going to test that hopefully soon in mice and see whether it works.
So, like, are you, like, more interested in, like, the physical therapy side of, like, a spinal cord injury or, like, perhaps, like, medication?
So, if ever we find a way to make the neurons that we're interested in respond to certain drugs, I think that would also be very beneficial, because the nice thing about drugs is that you take them and then it kind of spreads throughout the nervous system and affects everything. But, the problem with drugs is they often have many side effects. So either light or drugs or some whatever, I'm always impressed by what other labs come up with. Some people have thought about using magnetic waves or sound waves, right, to excite neurons. So if we're finding that that's effective, then perhaps we can make our neurons of interest be activated by sound or by magnets. So we're open to anything. Thankfully, there's a lot of smart people out there who are thinking of these things, and hopefully we can just kind of adapt our ideas to what we're trying to accomplish.
It's definitely made a lot of accomplishments so far, so I'm looking forward to the future.
Oh, great. Me too.
So for young scientists of the future, or people that are going to go into the neuroscience field, do you have any advice that you would give them about pursuing research, or just what makes this field so interesting and amazing?
Yeah, I mean, to me, the nervous system is the last frontier, right? Like everything we do, feel, our physiology is to some degree controlled by the nervous system, but it's also one of the least understood because there's so many neurons, so many different areas, but there's so many labs doing so many great things. So I'm really excited to see, like, every year I can't keep up with all the advances, which is great. So for young people who are interested in neuroscience, the great thing about the University of Ottawa and really the Ottawa region is there's many labs either here at the medical campus. Sorry, for those listening, we are right now on the main campus. At the medical campus, you also think of Carleton, you think the Royal Hospital, CHEO. There's a lot of really interesting neuroscience research happening and neurology research happening in the Ottawa region. So there's lots of labs looking for talented individuals to contribute in whatever ways. So it's just a matter of, it's not easy because there's also lots of students, but try to identify those labs that tackle research questions that are of interest to you, and kind of approaching the research supervisor and whatnot, and kind of making your case as to why you could be a good contributor to the research program.
So in terms of finding those opportunities, do you have any advice for students on how to go about looking for those, and like what they can do to like make themselves maybe more noticeable towards profs?
Yeah, I can certainly give you like what not to do, which is just a shotgun. Hey, I'm looking for a research lab, and that's it, right? I think that what will attract a supervisor to a student is them making their case as to why the research that the person is doing means a lot to them as a student. You don't necessarily have to be fully trained. No one expects an undergrad to have all the techniques and all the training, but is a passion there? And it's not always about the grades as well. I don't look at grades foremost. I look at the passion. I look at to whether there's a earnest curiosity about the nervous system. This is based upon jobs or sports or different factors that make me say, oh, this person might be interested in motor control. Or if you're interested in Parkinson's, there might be other ways to kind of sell yourself, let's say, to the study of Parkinson's through personal family history or whatnot. And then, if you have ideas, if you looked at someone's website and you say, oh, I thought this was really curious and I thought about this hypothesis or something like that, kind of give the impression that you've thought about why you are interested in their research and what you could contribute. I think those are good ways to go about it. And sometimes it can be in a volunteering capacity. So offering the information that, if you're looking for a volunteer, I am available during these times of the week or how often. The more information you give so that the person you're contacting does not have to get back to you and say, can you give me this and this and this? I think the more likely they will be looking at your email and thinking seriously as to whether you're a good fit to their research program.
I had one additional question. Going back to the very beginning, do you feel like your initial background in math and engineering helped set you up for success in the spinal cord realm now?
In the particular field that I'm in, which is electrophysiology, computations of the neurons, yes, it helped a lot because of the math background and also some of the computer skills. I cannot undersell how important computer skills are. Whether you're studying something that's computational or anything that has to do with data analysis or processing things or making things automated, having some familiarity with computer programming goes a long, long way. So it's one of those soft skills that I think makes you attractive to a lot of research opportunities. Whether neuroscience or different scientific fields, having that ability to process a lot of data, because more and more fields, people generate a lot of data, we don't know what to do with it, or it takes time to analyze a lot of data. So computer skills are really, really important.
It's good to know.
Yeah. I don't know whether I guess I should say that my work is supported by grants by the Canadian Institute of Health Research and the Natural Science Engineering Research Council. And once again, I've been blessed by great researchers, and so I'm just going to name some of them. So I've got a postdoctoral fellow named Alex Liberté, who's kind of really instrumental in kind of spearheading my DI3 neuron. That's the neurons that we're targeting in mouse research. And I've got a PhD student, Shahryar Naziri, as well as Dexter Lash-Burrows, who's an incoming master's student. So they've got a lot of exciting projects. And then in the zebrafish field, my postdoc now, she was a PhD student, Stephanie Gaudreau, instrumental in keeping my zebrafish program going. I would never recommend someone starting a lab and having two different animal models. I don't know what spurred me to do it, because you're kind of splitting your brain in two different directions. And like you said, they're not similar. So you have to kind of think in two different ways. But thanks to those people, then I've been able to have those two research programs and have them thrive. Thank you so much for the opportunity to share about my research program. Really appreciate the time you guys have had really great questions, and hopefully I've brought some insights to science.
Yeah, thank you so much for joining us today.
All right, that's a wrap. Thanks for spending time with us today.
We're your hosts,
Aditi,
Taryn,
and Amelia. We'll be back next time with more stories, insights, and a fresh cup of tea to spill.
remember, the journey doesn't stop here. Keep thinking, keep questioning, and keep steeping in inspiration.
see you next time.